So this is sort of the formation. The title of this is a bad title. Okay? This is not the polymerization of nucleotides into a long DNA strand. This is just the synthesis of our three components coming together through two condensation reactions. So my apologies with the title. Okay? But we can see here, so here was our site of attachment for our base. This one happens to be thymine coming in and bonding to this OH group on the C1, okay? And then this H and OH are gonna pop off and make a water. Over on this side, phosphate comes in, binds to C5. Again, we're gonna release another water. And if you already looked at the questions, I'm gonna ask you to sum up all the changes in all those bonds in this figure and spit out a change in enthalpy for me, okay? So we'll do that at the end. There's just another picture of it, okay? So now we're actually talking about polymerizing the nucleotides into a strand of DNA, all right? Like I said, that was a bad title for the other slide. Nucleotides are linked together through a phosphodiester linkage. Okay. The oxygen on the 3' is linked to the C5 of the next nucleotide. Okay, so we have a 5' end. Here we have a phosphate group. Okay, here's our C5 carbon. We have our sugar, and then the 3' carbon is attached to the next phosphate, and so we have sugar, phosphate, sugar, phosphate, sugar, phosphate, going from 5 to 3 is our backbone, okay? And coming off the backbone, the side groups of this whole chain, this polymer chain, are all our bases, right? Okay. I want to point out the name, because I will use this word, and you'll see it's in one of the problems, glycosidic bond, okay? Who knows a generic definition of a glycosidic bond? Generic definition of glycosidic bond is anything bound to a sugar. Its most basic definition. And so it could be a CO bond, or it could be the two things interacting. In this case, it's CN, right? It's a nitrogen to a carbon. That's why it's N, glycosidic. Okay, that's what we have there. And I only point this out because, again, I have brilliant students who have been in other classes and have everything memorized. And when I ask them, like, hey, how much energy does it take to break the glycosidic bond in DNA? And then they go pull the CO number off of the table. And they go, well, CO is a glycosidic bond in like a sugar chain. And I'm like, cool, but not in DNA. So, you know more than I need you to. You're so smart, but I have to mark you wrong because the glycosidic bond in DNA is between the sugar and the base, and it's carbon nitrogen. There were actually a lot of those on exam 1 last semester, surprisingly. Okay. So then the other thing here is this unit here, this phosphodiester linkage, okay? The two ester bonds are the CO bonds, okay? 2 of them, diester, CO. And then the phosphate in the middle represents the phospho part of phosphodiester, right? So we have, the way I like to think through this, I have C3 bound to an O, bound to a P, bound to an O, bound to C5 on the next nucleotide. This is ester, this is an ester right here. And I have these 2 other single bonds P and O. You're gonna get a question that's like, which one of these is the easiest to target? And then don't write, resonance from the double bond will make blah, 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 super hard. I didn't teach you that. That won't ever be the answer for me, okay? Keep it simple, right? Just knowing which bonds are which in basically, like I said, a simplified level. Glycosidic versus what's happening in this backbone string. Okay. So the oxygen that's hanging off of C3 will attack the alpha phosphate, okay, on an incoming nucleotide, right, at the C5. The phosphates are attached at C5. Okay. So when this OH comes in and breaks this bond apart, the pyrophosphate, the 2 phosphate ion pops off, and the linkage then forms here. And you also get water. I think the battery is dying in my mouse. I literally cannot use a trackpad. If I forget a track like my mouse, I'm basically useless. And I want it. There we go, cool. Okay, so here we have sort of our separate nucleotides, or we have a growing chain going on here. 5 towards the 3 end, it's growing in that direction. And then now we have all 3 of them hooked together in this picture. So chains are read from 5 to 3', and the incoming nucleotide gets added at the 3' end. And again, should be reviewed. You probably had to memorize in much more detail. This is as much as I need. Nucleotides are linked together through a phosphodiester linkage, producing the backbone. So when I'm talking about the backbone, I'm talking about phosphate sugar, phosphate sugar, phosphate sugar.